Good morning, everyone. So, how's everyone doing? Bharat, Atma, Utkarsha. So, hello, everyone. I welcome you all to the fifth session of this Data Science for Biologists program. Today, we are talking about machine learning. We will learn about how we can apply various machine learning techniques on trans- transcriptomics data to derive a biological significance from the data. We will also learn about regression, that is factor regression analysis. We will also learn about how we can apply differential gene expression analysis using DSEP on T-Bioinfo platform. And we will also try to understand what are the challenges associated with different types of the methods and what are the different considerations that need to apply before applying any method. So in our last session, we talk about metagenomics, but before that, during the penultimate session, on the third session, we learned about how we can apply, what is RNA transcription and what are the different methods by which we can measure mRNA abundance like RT-PCR, Northern blot, microarray. Then we will also learn about NGS RNA-seq technique, how we can measure RNA abundance using NGS technique. Next, we learned about what are different steps that are important for the RNA-seq data analysis. Here we will also learn about, we learned about what are the different algorithms that are used to pre-process and process this NGS RNA-seq data. Next, we demonstrated to you how we can generate a well-structured gene expression data table from raw RNA-seq reads. Then we also learned about different aspects of gene expression data table, that what it represents, like what are the columns and what are the genes. Then we also learned how we can visualize and interpret data once we have this structured gene expression data table. Next, we learned about how a box plot represents different summary statistics of a data. Then we also learned about various normalization techniques like log scale transformation. And we also understand how a normalization affects the biological interpretation. Then we also understand about quantile normalization method, and we understand how this normalization techniques affect data and how they make a data more normally distributed. Then we also demonstrated you that how we can manipulate our data and perform log scale transformation and visualize our data in the R. So Utkarsha, I think you just, if you can re-log in again, maybe that's why I'm not audible to you. So today we are going to learn about how we can perform various exploratory analysis once we have a gene expression data table. So to proceed, for those analysis. First of all, how we can identify differential gene expressions. Once we have gene expression table, so there are several factors that make it difficult to identify differentially expressed genes. So what are those factors? So there are thousands of the gene in each of these samples. Like we have 20,000 genes, or it could be a 7,000 genes. It could be a 60,000 RNA transcripts. Then sample in each group can have a significant variation in the expression label. And even when there is difference, we have observed it is hard to know what will be significant enough difference to say that are truly differentially expressed genes. So to overcome these challenges, we have to turn to statistical methods. So one way is to understand differences, statistical differences, that is t-test, that we have learned in our last session. So today we will understand how t-test can be applied only when there is normal distribution in the data. But like a gene expression data, which specifically read count values. So those read count values are non-normalized. So to identify the significant differences between non-normally distributed data of the gene expression, there are several methods like EGA, DSEC that are available in different R packages that can be handle that non-normalized data. So what DSEC do, that it uses a table of read count as the basis for differentially expression call, that is non-normalized counts of the reads. Then it uses negative binomial distribution to estimate the variance and mean dependence from read counts. So eventually it will compute p-value or p-adjusted value and as well as log fold chains. So today we will demonstrate to you how we can perform differential gene expression analysis using DSEC and what the output that we can obtain after running DSEC. So now let's go over the T-Bar-Info platform and let's see how we can apply DSEC. So meanwhile, let me just share the link with you. So once you are on this server, just let me know. So just type yes if you are on this server. Okay, Utkarsha. So if you have read count values directly, you have that table. So one way to apply DSEC, you just go under the data mining and you can try this differential expression. Today, I'm just taking a demo pipeline. So here under the transcriptomics, if you click on this, so again, we are considering that taking the same project, that is PDX project. So we are taking this third pipeline, that is demo RNA-seq. So once you're able to click on that third pipeline, demo pipeline, so with the start, we're able to upload the files. Then the next option that is getting highlighted, the Trimomatic. With Trimomatic, if you remember the third session, we provide you the details regarding how we can pre-process our data. So Trimomatic, what it does, it will remove adapters from your sequence. The next option that is getting highlighted, that is PCR clean, that it will remove PCR duplicates. Then we are taking Puchai 2T, that it will map, once you have clean reads, then it will align your reads on transcriptome. So you can generate read count using even the pipeline, that is HiSet and HDSet. Beside here, you can choose an option count values under the RSCM. So here we are able to produce both picm value as read counts. So then the next option is getting highlighted once you have gene expression table, that is DSEC2. So if you click on this option is under the differential expression. So if you click on that, we able to perform differential gene expression analysis. Then this end, with the end, we able to complete this pipeline. So how many of you able to run this pipeline? So we are running the almost the same pipeline, RNA-seq pipeline, only this time we are also considering a step for the differential gene expression analysis. So how many of you able to complete this pipeline? Just type yes. Is this some issue? So any one of you able to? Okay, Bharat. So okay, so once you're able to do this, just let me just repeat these steps. With the start, then you have to perform pre-processing step, that is PCR clean and trimeomatic. Then we are performing the mapping of the reads on the transcriptome, that is Buchai 2T. Then we are choosing to generate a gene expression table. We are choosing RSCM expression table. So here, okay. So then we can perform differential gene expression with the DSEC, and then we are ending this pipeline. So once we able to complete this pipeline, just download this first folder, that is DSEC2 all files zip. So if you download this, so here you will find various outputs. So let's see what these outputs contain. So the first output, if I click on this, what it contains, let's look at. So I'm just pasting it in Excel. So here what we have, first the gene ID, then the major important column, this one, ensemble ID or the gene ID. Then we have log fold genes. Then we have p value. Then we have p adjusted value. So these are the major and the important columns that we have to consider. So log fold genes will actually tell you that a particular gene with how much fold chains it is highly expressed or upregulated or the downregulated in condition 1 in comparison to the condition 2. So positive fold change means it is upregulated in the condition 1 in comparison to the condition 2. Negative fold change means it is downregulated. So 1 means it is 10 times highly upregulated. So you can take that cutoff. Then here is the p-value. So then we have p-adjusted value. Can anybody tell me the difference between p-adjusted value and the p-value? So anyone familiar with what is the difference between p-value and p-adjusted value? Okay, Bharat. So the p-value usually when you are taking two samples, paired two samples. So p-adjusted value when there are multiple testing, means you have large number of samples, then you are considering large number of samples. So usually whenever we perform differential gene expression analysis, usually we need to consider p-adjusted value. So you can always sort these genes. So you can set a filter, and then, so any one of you familiar with what was the p-value that is considered as significant p-value in the statistical test? So how much p-value is considered as significant p-value? So yeah, any p-value less than 0.05 is considered as significant p-value. So you can always sort your genes based on here. As there are multiple samples, so we can, smallest the p-value is the more, there is more significant differences there. So the p-value exactly, but it represents the probability of getting any particular result by chance. So if it is lower, it means only 0.05. What does it mean? That only 5% chances that you have obtained the difference by chance. So lower the p-value, there is highest the significant difference. So here you can filter out the genes first based on the p-adjusted value. If you still want to filter out your genes and sort your genes, so you can taken into the consideration of the log2 fold chain. So higher the log fold chains, both positively and the negatively, means there is more difference, or there is high upper regulation and the down regulation of that particular gene in condition 1 in comparison to the condition 2. So this is the first output that we will obtaining, and we will understand what are the important genes. So the next, if you look at the next outputs, so here the next output, that is GO terms or gene ontology terms. So I'm pasting it in another Excel. So specifically DSEC also provide us what are the important gene ontology terms that are actually associated with our significant genes. So which ontology those genes are associated? So like biological processes, cellular components, and the molecule functions. If you are biologist, you must be familiar with these terms. Gene ontology term, that is molecular function, biological processes, that is represented with the BP. CC means cellular component. So this is what the another output that you can obtain, that what are different genes, what are the different gene ontology that are associated with their significant genes. So the next output, this CAG pathway. So let me paste it here. So it can also provide us that which pathways are associated with our significant genes. So just in case while running the DSEC or DSEC2, if you are not obtaining this output, it means the number of the significant genes are not sufficient in your particular results. That's why you are not obtaining these significant pathways. So once you have significant genes, that is how much genes that are statistically significant in your conditions. So based on that, it will from those set of genes, it will eventually find out what are the gene ontology and what are the different pathways that they are associated with. So then the next output that here you are going to obtain, that is, let me just show this. So these are the MA plus. So you will understand how much genes are there that are actually coming after the log fold chains. So like here, this is the cutoff at the log fold chains less than 1 and greater than 1. So this is like it will represent you in this form of, in the form of plots. So here it is not one plot that we are not obtained, that is volcano plot. With the volcano plot, we will actually understand which particular gene we have obtained that is uh, above the cutoff, like more than one fold chains or the two fold chains. So we can get these kind of output. So beside, so these are the major outputs that you can obtain after running this DSEC pipeline. Is it clear to everyone? Okay, that's great. So you can explore yourself, and if you have read count values, then you can run your pipeline yourself directly under the utility section. So this is the example pipeline or the demo pipeline. So this is under the differential expression when you have your own read count data. So remember, DSEC and HR only can be applied when your data contain non-normalized read count values. So if you have FPKM values, which is further quantile normalized, then you can apply various simple statistical method like t-test, that is Wilcoxon t-test or the Welch t-test. So this DSEC and HR can be applied when you have read count values. So this is always very important thing to remember when you need to apply DSEC or HR, or when you need to apply simple parametric statistical test like t-test. So now let's move ahead with the presentation. So these t-test or the DSEC can be applied only when we have two disease condition, like you can say you have two cancer subtypes, like here in the PDX we are talking about ER positive and triple negative. So what we will do if we have multiple conditions? So to address such scenario, we have regression. So the regression analysis is a way of a mathematical sorting out which of independent variable have an impact on a data that we have, that is gene expression. If you look at this regression formula, it has both dependent as well as independent variable. So the dependent variable is related to the independent variable with the additional consideration that is constant and the intercept that is coefficient. So now let's understand this regression process with a simple example. Let's say we collected a data on the eye color, gender, age, and the height and the weight of the people. So we hypothesize that some of the variables here are related and some are not. We also think there are some exception to the rule, like age and height or the weight could be related, but not always. So the relationship observed in the data can show which variable related and which are not related. If they are related, how much or with how much extent they are related. So such a relationship can be positive, negative, linear, and non-linear. So linear regression is a way to further model such a relationship to determine a mathematical function explaining the relationship between a dependent and the independent variable in a data. If a relationship, uh, if there is a linear relationship, so such function is a predictive and will inform us how the dependent variable will change if we change this independent variable. So the overall goal of regression is to find the right coefficient of this formula that minimize the predictive error based on the data that we have. So one of the methods that apply this regression or that employ the regression, that is factor regression analysis. So this factor regression analysis is applicable when we have multiple conditions or multiple factors. So it is useful when there is variability in your groups or within group. So it allows analysis among those groups. For example, in this PDX project, we have two main cancer conditions. That is, we have two cancers, breast cancer subtype, that is ER positive and triple negative. But then within this cancer's condition, then we have two mice conditions, two types of mice, that is athymic and the nod. And if you want to perform analysis between the different cancer condition, but you want to filter out that those particular genes should not be related with the mouse subtype. So how you can perform that kind of analysis? The answer is factor regression analysis. So to understand it more, let's perform factor regression analysis using T-Barn4 platform. Meanwhile, let me share the data with you. So I'm sharing a link with you to perform factor regression analysis. So let me just show you what this data set contains before performing analysis. So if you look at this data, we have this ER positive samples, then we have this triple negative sample. So if you look at this within the ER, we have two type of mice models, where this particular cancer tumor is developed, that is nod, and other is athymic. So similarly in the triple negative, we have nod mice, and then we have athymic mice which lacks thymus. So to find out the differences only between the ER positive and triple negative condition by filtering out the another criteria, that this gene should not be differentially expressed in the mice subtype. This is what goal to perform a factor regression analysis when we have to consider multiple factors. So now let's go over T-Barn4 platform. So here under the data mining, we have this tool that is factor analysis. If you click on this factor analysis, so how many of you are able to download this file? So this is already quantile normalized data. Yes, so, that's great if you're able to download that. So now let's proceed with factor analysis. So, and from the data mining section, just click on factor analysis. So let me just take you again here. Here under the factor analysis, you just click on under the data mining, click on factor analysis. Then by clicking, the only difference in comparison to the other pipeline, here first you need to select all the filters before uploading a file. So by clicking on OK, we are going to start it. Then we have to select the factor analysis. So as this data is already normalized, so I am providing a data set which is quantile normalized. It is first log scale transform, then we have also performed quantile normalization. So we are taking it by default that we don't need normalization. In case you have only FPKM values, in that case you need to normalize your data. So then we need to add factors. So the next, it is asking how many factors are there. Factors means how many first conditions, like we have ER positive and triple negative. So here we are selecting those conditions. That is, A means ER positive, we are going to select. Then B means that is the other condition, that is another cancer subtype, that is triple negative. Then it is asking how this number. So if you read it here, please add the factors and the number of the labels in each factor. So then in a particular condition, how many further labels are there? So as we know that we have two mice model, so we are giving two labels. So depending upon how much further labels or factors are there, you can add this value. So then we go to the next. Here we need to provide that file. So the next is check. Click on the check. So now we have to drag our samples in different factors. So this is the triple negative thymic into the one group. Then triple negative nod into another group. Then ER thymic into this group, and then we are ER with the node mice, we are giving it to the another group. So we have now 4 groups. So eventually A1 and B1 on what it represent? A1, B1 and A1, B2. This is ER positive group. A2, here we are in the A2 and B2, we are representing our triple negative samples. So with the continue, then and this pipeline. So by clicking, then next, after naming, we need to click on run pipeline to complete this pipeline. As this is a real pipeline, it's not a demo pipeline. So it will take 4-5 minutes to complete it. So how many of you able to complete this pipeline? Okay, Bharat. So then you can check the progress of your pipeline. So just clicking on my pipeline, still this pipeline has 0% progress. It will take 4-5 minutes. So apart from Bharat, who is able to completely run this pipeline, so it will take a bit time. You can refresh it, this pipeline, to check the status. So apart from Bharat, who else able to complete this pipeline? So I can always refresh my pipeline to check the progress of pipeline. Like it is already progress the 66%. Okay, that's great. Okay, so now my pipeline gets completed 100%. So now I can check the output. So here I work by clicking on, then it is download pipeline output. Here I have obtained this output. So now let me just copy it and paste in the Excel just to show you what it represents. So if you just look at the first column, that is gene. So other are the expression table, and the last three column. So this one, then sorry, I need to color it different color, and then this one. So what this column represent? So let me add a filter even on this as well. So if you look at the last column, what it represents, that it is giving you p-value using a chi-square test and b1, b2 versus a1, a2. If you remember what are our a1 and b2, that were our cancer subtypes. So the last column represent the genes, the p-value for the genes that are significant, that are differentially expressed between conditions, first two conditions, that is ER positive and the triple negative factors. So that is cancer subtypes. Thus, if you look at this column, what it represent? P-value for the B1 and the B2 and A1, A2. So what are our A1 and A2? That is mice subtype within ER positive condition, and these mice subtype that are in the triple negative conditions. So if you want to filter out only the genes that should be significant differentially expressed in the cancer subtype but not in this, so how you can filter out? So first just filter out the smallest p-value. So now you have to consider only those genes that are significant only in the third condition, but those genes should not be significant differentially expressed in condition 1 and the condition 2. So for example, in the first gene, if you look at here, we have p-value less than this 4.4, that is minus e, that is epsilon power minus 153. So it is significant in this cancer subtypes, but it is not significant in this. While if you're looking at the second gene, it is found to be significant differentially expressed in all the three conditions. So we cannot consider that it represents the only difference. This difference is only due to the cancer subtypes. So this difference can be because of cancer type as well as because of the mice subtype. Similarly, if you want to consider only those genes that should be only significantly differentially expressed in mice subtype but not cancer subtype, so you can filter out the genes that is significant differentially expressed in condition 1 and condition 2 but not this condition. So then we can let's take some genes, like if I'm proceeding, okay, if you look at this particular gene, so it found to be significant differentially expressed in these two conditions, but not in this. So you can filter out all those genes based on the required condition that is found to be significant differentially expressed in the desired condition. Is it clear to everyone how you can interpret this output? Is it clear? Okay, that's great. So now let's move ahead. So this is what it output represent, the factor A, factor B, and the factor C. So if you have any question till now, before heading into the next part. So far, we have discussed the methods that we can apply if you want to consider one variable at a time consideration, or what you will do if you want to take multiple features into the consideration. So if you want to take a multi-dimensional data, so and you want to consider that you drive your biological interpretation not based on one or the two genes, right? So if you want to drive the meaning based on the multiple and want to combine all those multiple features to derive a biological significance, you can. So the one method with which we can derive a meaning, or we combine a multiple features or multiple variables, so that is answer to that is machine learning. So what is machine learning? Well, many people will think about formula, or maybe some code, or maybe some flowchart algorithms. For the computer scientist or mathematician, these algorithms are definitely relevant, and that requires a depth knowledge about these domains. But here we interested in how a machine learning can help us to study big RNA-seq datasets. So what we will try to do is to limit the technical details of each of these algorithms, and instead we will create a logical series of the steps that will illustrate how the methods are useful, as well as what they might offer to study a biology in the RNA-seq data. So now question arises, why do we need to apply machine learning on RNA-seq data when we have number of statistical methods? So the answer to high dimensionality of the data. So what does mean high dimensionality? That there are, like for a gene expression table, we could have a 20,000 genes. We could have a hundreds of genes that are important for the consideration. So we can apply statistic only when we have small number of variables. So how we can analyze and visualize such a multidimensional data using the information from multiple variables, say 100 or the thousands of the genes. So to answer to such question is machine learning. So what are different types of the machine learning? There are two major categories. First one is unsupervised, second is supervised machine learning. So now let's understand each of these methods one by one. First, unsupervised machine learning. So an unsupervised machine learning focus on the detection of the pattern in the unlabeled data. So unlabeled data mean we do not have information regarding the class or the groups of the samples. So what does mean by class? That is, we have no idea about like what is the cancer subtype, So here the main idea is to apply techniques that are automated and relies on various assumptions about the data to learn about it. So what an unsupervised machine learning method try to do, or an algorithm try to do, that it will try to find out the patterns within the data itself. For example, here we have this squares, we have this circles, so then we have this pentagons. So this algorithm try to understand these patterns. So then it tried to find the similar groups together and try to group each of these groups. For example, here, what the result that you will obtain, that it will try to find what are the similar data points. Here it will cluster or group these samples. Like here, it will cluster the squares, it will cluster this circles, and then it will also pentagons. So this is how this unsupervised machine learning method try to do without having the knowledge of any prior knowledge of any of the label or the class of these samples. So it is only try to understand the patterns within these data points. So what are different unsupervised machine learning algorithm? It could be a clustering method like hierarchical clustering or k-mean clustering, then principal component analysis. So then we have also the supervised machine learning, which take into the consideration the classes of the sample. So what are the main applications of this machine learning in the biomedical data? So for the clinical purpose, especially in clinical research, it's used to study disease mechanism, treatment selection and development, as well as the predictive model can be very helpful. In our course, we will use machine learning method for the patient stratification, disease classification, diagnostics, predicting the response or the lack of there to understand the mechanism, both disease and the onset. So these similar methods can be applied to the different pharma and biotech industry for the biomarker discovery, then detection of the toxicity at the in vitro stage. Then it is also used to analyze the molecule mechanism of a drug action during the target discovery. And these machine learning methods are widely used for the drug repurposing or drug repositioning. So before understanding into the one of important unsupervised machine learning algorithm, that is principal component analysis, let's understand what is which is dimensionality reduction method. So no, let's understand what is dimensionality reduction. So first let's talk about dimensionality. What does it mean? If we look at our table of expression, we will see that it has thousands of the genes, that is rows, and several columns for the samples. If we take the first two genes, for example, in this table, we have 8 samples. We can visualize the sample in the two-dimensional space by taking the expression of the two genes across 8 samples. So here in the this plot, genes are the dimensions, that is representing x-axis and the y-axis, while sample represented the objects or the data points. So to study how gene expression can be used to measure the variability between the samples, we can use a dimensionality reduction technique, that is PCA. So now let's see if we add another gene like here. So we can view this as a three-dimensional space, but we have thousands of the genes or the multi-dimensional. So what is the different methods, how we can visualize all the features? So that is why we are going to use the PCA. that compress the data from the large dimension of the variables into the small number of variables that will help us to make the sense of the data. So let's try to understand how PCA works. So if we are using the approach of principal components, we looking for such a plane or the vector in the multidimensional space that we cannot explain the variance in our data. Here is a data with the low variance and we have a data with the high variance. However, this variance is not explained well by x-axis or the y-axis. But you can draw a line in the original space that shows the maximum variance of the data. So this vector through the multidimensional space can maximize the variance of the data is called principal components. And just like previous examples, we can add another principal components here. We have PC component 1 and PC component 2. If we select the principal component that explains the max variance of our data, we can project our samples into this space and visualize our full data. So this technique is called principal component analysis. So one of important point to remember about principal component that here each first component explains the maximum variance of the data followed by the other components like PC2. So here PC1 would be the component that will explain maximum variance in your data when you present your sample in a space. So far, to understand principal component analysis, let's perform PCA on tape info platform to understand the better regarding the PCA, what it represents and how we can visualize our data. So as we know that from the gene expression table, we have thousands of the genes. So let's perform PCA. Sorry. So let me share the same gene expression table with you that we have obtained after we have FPKM values. So today we also learn how we can perform log scale transformation and quantile normalization on T-Info platform. Then we can apply how we can visualize our data using PCA. So I am sharing the same data that we have obtained after running the gene expression pipeline. Just let me share the link with you. So this was the gene expression table that we have obtained after running the RNA-seq pipeline. So last time we learned how we can perform log scale transformation in the Excel. So now we can also look at how we can perform normalization, first normalization using T-Info platform, and then we can also apply PCA. So once you're able to download this, here under the utilities section, once you're able to download this file, just let me know that. Okay. So here under the data mining, if you go to the utilities section, just click on utilities. Here you need to provide the file. Okay, so okay. Okay, so I'm going again. Thank you, Bharat. So under the utility, under the data mining, we have this section that is utility. Is it now, my screen is visible to you? Okay. So under the data mining, we have utility section. So then once you click on that, then we are getting this option file upload. So we are uploading the same gene expression table that we have obtained after running the RNA-seq pipeline, which contain FPKM value. So here what we are going to do, first we are going to perform normalization on our data, then we are going to generate PCA components. So here under the add files, just upload this file that I have provided you. So once we able to upload your file, then just click on continue. Then the next option, that is start. So the next, sorry. Next, we are under the utility, the first option we are going to perform quantile normalization. So here what this pop-up will give us, it asks us to select the parameters. The first parameter that it is asking, that is transform table to ln. What ln means? Log scale transformation. As this data is contain only FPKM value, so first of all we are going to perform log scale transformation. So I am going to do this yes. Then we have to select the threshold, that what for the quantile normalization, how much threshold we needed. So after the log, if you remember last time, when we have log normalized our data, so the minimum and the maximum value that we obtain, so that is 0 to maximum 20 value. So we are filtering here, we are selecting this threshold 5 to filter out those genes which have expression more than 5. So the next, once we able to perform the quantile normalization, next option that is getting highlighted. For example, here if you want to do only quantile normalization of data, you can directly end this pipeline. As here we want to visualize using the PCA, so we are selecting this option. So it will ask next whether you want to select a filter threshold. So I'm taking by default option. Already we have log transformed our data, and we are going to use standardized PCA, and we are going to standardize our table column. So I'm here taking all the by default option. You can play with this option and can change it as well. Except that if you have not normalized your data, so in that case you need to you can also perform log scale transformation. So I'm here taking by default options. Then we are going to end this pipeline. So we have performed quantile normalization, then PCA pipeline. Now save it and run this pipeline. It will take a bit again 4-5 minutes to complete this pipeline. So how many of you able to run this pipeline? So Bharat, what's the problem that you are facing? So it's a complete, okay. So can you tell me exactly what the problem that you are facing? So I can show it again. So just I'm selecting this. Okay, can you restart it? Otherwise, what you can do if you're still not able to do this, just need to upload your file. You can restart it. Then click on start. Okay, so I'm repeating it. So with the start, by clicking on start, then we are going to perform quantile normalization. Here we are selecting first transform table to the ln, that is we are doing yes log scale transformation as we have this fpkm data. So if you have already log transformed values, in that case you don't need, then we are selecting a threshold 5. Once we able to perform quantile normalization, next is pca. So here in the pca I'm taking all the by default options, I'm not changing, but you can play with that later. So then we click need to save that and then end pipeline. So I'm not running this pipeline as I have already run this pipeline. So I can skip that. Is it clear to you, Utkarsha? And Bharat, whether your pipeline get completed or whether you're able to load pipeline or not? Okay, that's great. So Bharat, if you're not able to do this, I think we will share this video with you later and you can. Okay, so might be there is some problem with your might be the account. So we will check it and you can run it later. So meanwhile, let me just go to the my pipeline. So here my initial pipeline already completed. So here we have two outputs. The first one is the result, PCA results. So I'm just downloading the first output first. And the second output, it is quantile normalized value. So let me just show you. So this is so these are the PCA results. The other output is quantile normalized. So right now I'm taking this PCA because we have done the normalized last time. So right now I'm skipping that. So you can compare non-normalized data with the quantile normalized data as well. So here, but it represent. So here these percentage, what it represent each of these components. So this would be the first PC component, then this is PC2 component, PC3. So from thousands of the genes, what we are reduced, so how many output PCA component that we obtain, that is near to 10, this 10 or 15 components, right? So this is what dimensionality reduction means. So here how we, so what these components represent, how we can visualize our data using these components. So if we're taking these two and let's plot a scatter plot. So if you go to the scatter plot, so from this scatter plot we are unable to understand exactly what they represent, so we can do the modification to make what these each of these samples they are. So how we can do that? Just click on this, select the data. So here we can add the labels, add a new series. So here we know that we have two type of sample, that is ER positive. So ER positive sample, then we have what are those ER for the X values. So X axis, what it represent. So if you look at this ER positive samples, these are its X axis. Then what are this samples, Y axis. So we are considering the second PC2 component. So we are going to plot PC1 versus PC2 for the ER positive is selected. Then do just OK. So then add a next for the triple negative samples. That is TN negative. Then for the series x-axis, what it represent? If you look at here for the triple negative x-axis, sorry. Then for the y-axis, so here for the triple negative sample, so we can select there these triple negative for the PC2 components. So as now we can delete the initial series. So just remove that and done OK. So now you can clearly visualize based on the, now we can also draw what is our x-axis and the b-axis. Just go to the layout and just define axis titles, primary horizontal. So what is our x-axis? That is PC component 1, PC1, that represent how much variance? That is 17.79. So the first PC component represent the nearly 18% variance in the data. Then what is this y-axis represent? Vertical axis. So this represent PC component 2 with how much variance? That is 7.36. So now we can, based on the two components PC1 and PC2, we can visualize our data, visualize the pattern within our data. So based on two components, we can clearly see that ER positive samples are different from triple negative samples. So even some of the here, if you look at these samples, so most of triple negative samples are aggregated or clustered together. Only this and this particular samples is getting actually a bit, might be act as a outliers. Is it clear to everyone? So this is how, whenever you have a data, first of all, to understand how much, what could be the possible patterns within your data, you can always go for the PCA. Is it clear to everyone? How we can visualize the pattern of our data? So in the transcriptomics 2 course, you will find a video how you can plot this scatter plot once you have PCA components. Is it clear? So now we can clearly see that there are two types of samples that are different from each other. So any one of you able to produce this scatter plot based on PCA components? You can also then give a title. Sorry, chart title. So what it represent? Scatter plot. Based on PCA components for PDX data. PDX gene expression data. So any one of you able to produce this PCA plot or scatter plot? So any one of you able to produce PCA plot or not? Except from the Bharat. So you need to select the first two components. Okay. So these videos can be shared with you and you can draw these plots yourself as well later on once you have this data and you understand what it represents. So anyone, Utkarsha, Atma and Santesh, any one of you able to produce PCA, this scatter plot or not? So the importance of the PCA is always, it will let you understand what are the different patterns within your data. So just if you draw, okay, Santesh, so you can do it later. So we have just renamed this data based on this variable. Already sample names are there. So based on, we know that these are ER positive and triple negative samples. So based on, we just rename those samples. So how you can do that? Just click on that and select data. Okay. So okay, now let's move ahead. Importance of the PCA, whenever you have any gene expression table, gene expression data, to understand what are different patterns. So always first step that you need to do once you have gene expression data table, that you must check the and visualize your data. So PCA is one of the best method. So this is like how you can directly when you can do it in Excel. Beside on T-Bind4 platform, if you provide the data here, let me just, we have another option that is unsupervised analysis. So in the unsupervised analysis, you can also use PCR library. So it will directly give you that output. So if I can do this, let me, meanwhile, I will go to the next part. So I can just show you. You just, if you select this unsupervised machine learning, just select unsupervised machine learning and just provide the same gene expression data. So this is the another method. So this is the same thing, only here you need not to draw plot yourself. So then you can do continue. Just start. So just go to PCR library. So here you need to select number of PC components, the maximum 3. Then if you want to scale the data, center, transpose because we want to do the, make the PCA components of the genes, not the sample. So I'm doing this by default. So then you can do the end. So this is PCR library. Here we have not used normalized data, but you can take the normalized data. So I'm just showing that there we have another option. PCR plots. So here we are selecting only maximum 3 components. So in the now, while this pipeline is running, let's move further into the session. So another unsupervised machine learning method, that is hierarchical clustering. So what is hierarchical clustering? That it is an algorithm that groups similar objects into the group called clusters. So hierarchical clustering can be of two types, that is agglomerative and the divisive. So what is an agglomerative approach? That initially each data point here is considered as indivisible cluster. So it starts with the step 1. Here each of the data point or the sample is considered as an indivisible cluster. So at each iteration, similar clusters are merged together to form a new cluster. So you compute the proximity matrix, let each data point to be a cluster and repeat this step until you're able to obtain a single cluster. So this is how agglomerative clustering works. So it's starting from all the data point to make a single cluster. So the divisive approach is just reverse of that. It is consider all the samples first clustered together and then it start to divide each of these clusters. So that output that we will obtain after running the hierarchical clustering, that is cluster dendrogram. So what this cluster dendrogram represent, that if you look at here, so here you have minimum number of the cluster, at the bottom you have maximum number of the clusters. So to understand it better, let's run hierarchical clustering on your data. So let me just share the data set with you. Even you can take the same data. So only that we have used for the so we can take the quantile normalized data for the factor regression analysis. So I am just sharing the same data that we have quantile normalized. So the data set that we used for the factor regression analysis, we can use the same data here as well. So again, I'm just sharing link. So to perform, so that is already normalized data. So if you go to the area of analysis and go to unsupervised analysis. So this is the same data that we have used for the factor regression analysis, which is FPChem quantile normalized already QN5 data. So the file ends with QN5. So if you do upload files, so after start, then we can select if under the going under the clustering. So here you will find k-mean clustering, different clustering methods. So here we are selecting H cluster, which is hierarchical clustering. So after click on hierarchical clustering, here you will find different parameters. So here you will get like average. So if you click on this, you have different methods, var, d2, centroid, complete, single, and average. You can find details of these methods in the transcriptomics 3 course. So these are the different linkages. So we are selecting the average, and then it is also asking what is distance metric that you want to choose. So here the different methods like Euclidean. So what is the distance metric? That how you will find the dissimilar or similar data points. So we are selecting Euclidean. Then how many clusters you can select the number, how much cluster you are expecting. As we know that in this particular data that we have used for factor regression, we have minimum 2 clusters, that is ER positive and triple negative, and maximum we know that there are my subtypes as well, so I'm selecting 4. Then whether you want to do transpose, I'm selecting true, as we want to cluster the sample, not the genes. So we are selecting these first two default, by default options. We are just choosing the number cluster 4. So even if you don't know, you can play with this number of the clusters. Then we are doing a transpose of data, as we want to cluster the sample, not the gene. If you save this, and here is hierarchical clustering, H clustering. Even my PCR plot library already completed. Now we can look into the output of this file right now. Meanwhile, so this is what the PCA plot that we have obtained. So you can, because that data is not normalized, so this is how these PCA plots actually generated. So you can filter out, you can also group these samples. So although here there is no much separation because this data was not quantile normalized. So you can, with the PCR library, if you provide the already normalized data, so then you can make directly draw these plots. So any one of you able to run this hierarchical clustering pipeline? Still processing. So any one of you able to run this hierarchical clustering pipeline? Okay. So my pipeline already get completed. So now let's look at the output. So first I am just considering this output, that is PDF file, right? So if you look at this now, we have obtained something like this, a dendrogram. So here what we are getting, like this triple negative, we are getting a cluster of this triple negative, one of the ER positive sample clear to near to that. And if you look at these, all ER positive samples are actually clustered together, and two of that athymic. So if you look at these athymic ER positive sample are clustered together, and if you look at this triple negative NOD samples are getting clustered together, and then this ER NOD and ER NOD, this sample is although these clustered with this ER positive, but a bit different from athymic. Similarly, athymic triple negative samples are clustered on this side. You can see the pattern based on the all the genes. So these samples can be even clustered even better after selecting a particular significant gene. So you can always cluster and play with that, and you can also play with the different parameters. Any one of you able to obtain this output? Beside this cluster dendrogram, we have these two cluster table clusters only. So let's download what we have here. So let me just paste it in the Excel. So here what you are getting, that is cluster ID. So you can also find out exactly apart from that dendrogram. So here we are also getting information like this particular is clustered in the cluster 1. Then this is cluster 1 samples, ER positive, and these are clustered 1. Then this is what the second sample, this is cluster 2. So you can find out all those details. Is it clear to everyone? So these are the unsupervised machine learning method. We have then k-mean clustering. What is the difference between k-mean clustering? The if I'm in terms of output, you will get this kind of output. In that case, you are not getting dendrogram. But usually from the dendrogram, we can clearly visualize our sample. So that's why a hierarchical clustering a bit a better way to represent your data, to understand the patterns within your data. So now let's move ahead with the supervised machine learning. So this is for the hierarchical clustering, and this is what the output in the form of a dendrogram. So as we go from top to bottom, the number of clusters increased. At the top, number of clusters are minimum. So the next is supervised machine learning. So as its name indicates, so these methods rely on the availability of the training data that has been labeled with the class labels. So a supervised algorithm learns the information from the training data. For example, here, so then once it learns, so then it applies this learned or supervised knowledge to analyze the new data. So these are the labels. So usually this information is available in the training. So the algorithm learns class or the labels of samples from the training. Once it learns, then it applies its learning on a new data to predict the label of those classes. So in the biomedical project, there could be a clinical data or other phenotypic data like type of tumor. So here we know that it will explain like what is square, what is circle, and what is pentagon. But in medical data, these labels can be cancer or the normal or cancer subtypes. So once the training or the learning is established, so we call those models as supervised machine learning models. So the no new data will be assigned with the class that is present in the training data. So what is the important aspect? Your features or the genes should be the same. So now what is the training data? Overall supervised machine learning learns based on the training data having labels. So then these supervised machine learning can be of mainly two types, that is classification and the regression. In the classification, you are actually classifying two classes of the samples, while in the regression, it indicates the continuous values. So it will predict the continuous value, while in the classification, you are actually dealing with a particular category or the discrete values. So what are different examples of supervised machine learning? That is, could be decision tree, random forest, sport vector machine, linear discriminant analysis, logistic regression, naive Bayes. So these are some of the examples. There are nearly 20 methods are available that are supervised machine learning algorithms. So now, so what is the training data? Here in the training data, we have this class. So it class can be As I have told you that it could be like we are talking about ER positive, triple negative, then we could have cancer or the normal. While the test data, it lacks that class. The only difference between, so in case of unsupervised machine learning, you need not to provide the label, class label. While in case of supervised, for the training, you must have a class. Then based on the class, then it will predict the labels of the test data which is unknown. So in important aspect regarding whenever you're dealing with the machine learning, there are some terms like features. So the features means the genes, and then samples in the machine learning, we call them as objects. So one of supervised machine learning method is random forest. So it is a well-known supervised algorithm, which is a natural extension of a decision tree method. So what is a random forest? That it is a technique that automates decision tree using a process of random initialization, that it randomly select a subset of a data and then do the voting. So here each tree is going to predict the class of label. For example, here once it produce number of trees, that is random number. So each tree predict a class of a label, like tree 1 predict class A, tree 2 predicts class B, and tree n predict the class n. So class B. So eventually final class of the sample is decided with the voting method, that if the majority of the trees saying that this class have this particular label, so that label of the class is assigned to the sample. This is how a random forest is work. So this is the binary decision tree. So we are here it will define based on an expression of a particular gene sample. It will classify a sample into the class 1 and the class 2. Then it will take consideration, it classify that sample. Then based on the next expression of the next gene, it will classify these sample. So this is how a binary decision. Binary means it classify the samples into the two classes, whether it's class A or the class B. So it is how binary decision tree works. So for the supervised machine learning, another important aspect is feature selection. So what is feature selection? That is, it is a process of reducing the number of input variable when you're developing a predictive model. So it filter out important genes, that it rank the genes based on the performance of each of these category. So these feature selection method can be a filter based feature selection method, it could be a wrapper based and intrinsic feature selection method. So specifically random forest and stepwise LDA are two of the machine learning algorithm that can also can build a prediction model as well as can perform a feature selection. So we call this method as intrinsic feature selection method. So the difference between feature reduction and the feature selection, feature reduction is all take all the features and then generate a new features or new dimensions. While in the case of feature selection, it only rank important features. So you can understand what are important features in a particular data. So I think we don't have much time to perform the this supervised machine learning algorithm. So we can try it in the next session when we are going to talk about project designing. We can run a supervised machine learning algorithm, particularly random forest. So if you have any questions so far for today's session, so you can ask before we end this session. So you can run all these pipelines yourself once you have this video. So we are going to run this pipeline next time. So there is some these assignments in the next session. Complete the transcriptomics 3 course, and 2 and 3 complete both the courses, where you will get in the 2 you will get all the statistical analysis, and in the transcriptomics 3 we have covered most of machine learning methods. So then you can also look into the this code OmicsLogic platform to run and understand to perform PCA and all those hierarchical k-mean clustering and hierarchical clustering. So in the next session, we are going to talk about how we can proceed to plan a bioinformatic research projects using dataset from public repositories. So if you have any questions for today's session, you can post your questions in the chat box. So if you have no questions, so thank you everyone for joining today's session, and we will meet you the next Tuesday.